I'm Dr. T and welcome to my office. So today I'd like to talk about nucleic acids, and in doing so I first need to talk about their monomer, because nucleic acids are polymers and their monomer is going to be the nucleotide. Nucleotides are comprised of three constituent parts. The core of them is going to be a 5-carbon sugar known as ribose or deoxyribose. The difference between the two is the missing oxygen. The carbons on the carbohydrate are denoted by a number followed by a prime sign or an apostrophe. This is to distinguish them from the carbons in the rest of the nucleotide. So going around the ring, we have the 1' carbon. This is where another part of the nucleotide is going to attach the nitrogenous base, which I will get to here momentarily. On the 2' carbon, we see the difference between RNA and DNA. In RNA, this is going to be a ribose, and there is an alcohol on that carbon, as you would expect for most carbohydrates. For deoxyribose, that alcohol is replaced by simply a hydrogen. Therefore, there is no oxygen there, hence the deoxy part. This improves the stability of the DNA nucleic acid, as there is a degradation form that involves reaction with this particular oxygen. By having it absent, it leads to a more stable polymer. The 3' carbon is going to be where nucleotides attach, or at least I would say where the next nucleotide will attach to. The 4' carbon is part of the ring, and its alcohol is what is forming the oxygen in the ring. Typically speaking, it doesn't get much press. The 5' carbon is going to be attached to a phosphate group, which itself may likely be attached to multiple other phosphate groups. The phosphate groups provide a couple of functions. The phosphates will be connected by phosphodiester bonds. These are fairly unstable, and when hydrolyzed can generate a fair amount of energy. This allows nucleotides to be an energy transport molecule and one of the primary short-term energy transporters within the cell, what most enzymes will actually utilize in order to function. You can have up to 3 phosphates on here. If there are 3 phosphates, then we call this a triphosphate. If there are 2, it's a diphosphate. And if there is 1, it's a monophosphate. If you are familiar with adenosine triphosphate, that is a 3-phosphate nucleotide. Now, moving back to that nitrogenous base, this is what gives each nucleotide its identity. There are 5 nitrogenous bases, all of which are aromatic ring or rings that contain nitrogen within them. It's important to highlight here that while all benzene rings are aromatic, not all aromatic rings are benzene. Additionally, not all aromatic rings contain just carbon. They can contain other elements such as nitrogen, and these do. There are 2 types of aromatic ring structures that are utilized by the nitrogenous bases. The purines, which are actually a pair of rings that are conjoined, and the pyrimidines, which are a single 6-membered ring. The chemistry of these rings isn't dreadfully important. However, what is important is their intermolecular forces and how they interact with other individuals, whether those be enzymes or with other nucleic acids. There are 2 purines, that of adenosine, otherwise known as A, and guanine, otherwise known as G. Adenosine can form 3 carbon bonds and will do so to the pyrimidines, either thymine or uracil. These two are very, very close, and the distinguishing features between them is one that does not affect their interconnectivity, but does affect their ability to be recognized by enzymes as being different. Thymine, aka T, is used in DNA, and uracil, aka U, is used in RNA. The thiamine is a little bit easier for error-correcting enzymes to recognize if there is an error, and thus it is used in DNA. The last pyrimidine is going to be cytosine, aka C. This forms 3 hydrogen bonds and will do so with guanine, G, thus making our base pairs C and G, and A and T, or A and U, depending if you are in DNA or RNA respectively. Now, when referring to nucleotides, typically we distinguish these by a 3 or 4 letter abbreviation. If they are an RNA nucleotide, which is by far the most common within the cell, then they are distinguished by a 3-letter abbreviation. The first letter is the letter of the nitrogenous base. So if it's adenosine, it's going to be A. The second letter indicates the number of phosphates. So if you have 3 phosphates in phosphodiester bonds on the 5' carbon, then it would be tri or T. And then finally, the last letter is always going to be a P for phosphate. So adenosine triphosphate, aka ATP, is going to be a nucleotide. Now, if this is of the deoxy variety, so it's missing that particular oxygen, then a lowercase d will be put in front of it. So DATP would be the deoxy version of adenosine triphosphate. Once again, it's actually only different by a single oxygen. As far as uses for the nucleotides go, there's actually quite a variety. For the deoxynucleotides, it's basically simply as a monomer for DNA. DNA doesn't get used for much except for long-term data storage, which of course it's very, very important for, but all of the other roles for nucleic acids tend to go to RNA. For the RNA nucleotides, one, they can be used as a monomer to form RNA oligomers and polymers. Alternatively, though, they can be used individually. As alluded to earlier, adenosine triphosphate, as well as a few of the other nucleotides, but specifically adenosine triphosphate, is used heavily as an energy storage molecule, a very short-term energy storage molecule, but an energy storage molecule. Typically speaking, energy will be extracted from other biomolecules such as carbohydrates or lipids and converted into adenosine triphosphate. I should say the molecules aren't converted. The energy is taken out of that, and that is used to assemble the phosphates on adenosine triphosphate. Then enzymes can slice off 1 or 2 of the phosphates, releasing energy during the hydrolysis of those phosphates, and use that to do whatever they need to do. Other things such as GTP are also used for this function, but far less commonly. Additionally, you can use the nucleotides for communication molecules. Cyclic AMP is a classic case. In this case, the phosphate is rearranged, so it's actually forming a second ring within the structure. And this is used to communicate as a signal between different enzymes. Its production is triggered by one set of enzymes, and then other sets of enzymes are triggered by its presence. So with that said, that's nucleotides, and in later videos I'll jump into the polymeric forms. So talk to you later, and have a wonderful day.